excess stain. When you rinse away the excess stain, you don't want to wash your bacteria off. So that's why you have to heat fix for a direct stain, because there's rinsing involved and you don't want to lose your bacteria. You want to stick them to the slide. When you do a negative stain, you're going to add what kind of a stain? An acidic stain. And you're going to add that to the slide and mix your bacteria into it. And what color are the bacteria going to come out? Good answer. They're not going to come out of color. They're going to be clear. So when you use a negative stain, your bacteria are going to be clear. It's the slide that you're staining. You're staining the background. So now think about a capsule stain. Remember, you saw your little cell was stained purple. Then you had a clear area where the capsule was, because the capsule repelled the stain, right? It was white or clear. And then your background was stained. A capsule stain is actually a type of a negative stain because the capsule repels the stain and comes out clear. And the whole background is stained. Okay, in a negative stain, you don't heat fix. Why? Why not?
Distortion.
What?
So you don't distort it?
Okay. Because you don't heat fix, you're not going to distort the shape of your cells as much because you're not melting them. But why don't you heat fix when you're doing a negative stain?
Because it could damage the bacteria.
No, because you're not going to rinse. In a negative stain, you're not going to rinse. You're going to put the stain on the slide, mix your bacteria into it, and leave it. So you don't rinse a negative stain, so there's no need to heat fix, which turns out to be a good thing. Because if you want to see your cells without distortion, that's the way to do it with a negative stain. Thing is, negative stains don't work for all types of bacteria for whatever reason. For example, Treponema pallidum. That one's hard to stain and won't take a negative stain. They do a special silver stain with Treponema. Okay, and let's see. When you did your flagella stain, it was 2 steps. First step was tannic acid. Second step was carbofusion, pink, right? Why did you use the tannic acid? To make the flagella bigger so you could see them. To thicken the flagella. Yep. Tannic acid is a mordant. It's sticky. It sticks to the flagella, makes them thicker, so they look bigger. It's easier to see. Good answer. Okay, we discussed this in lecture just today. In the gram stain, when I write gram on my paper, I have to use a capital G. Why?
Because it's a name.
It's a name. It's a formal name. It's a guy's last name. Good. Okay, the acid-fast stain and the gram stain are both what kind of stains? Differential. Good.
Which gram stain?
And the acid-fast stain. They're both differential stains. Which reminds me, what was the name of the acid-fast stain we just did? TB. Who?
I forget.
Quinone. The TB quinone stain is the type of acid-fast stain we did.
Wait, what's the name again?
TB quinone. It's in one of those documents in Canvas. In the acid-fast stain document in Canvas. It might be in both documents. TB quinone. TB stands for tuberculosis, and quinone is the man's name that invented it. Okay, you need to know your structures, what they're for. What's a flagella for?
Movement.
So it can swim. The bacteria can move. Movement. What is a capsule for?
Protection.
Protection. From what? From phagocytosis. Protects them from being recognized, and also helps them slip away. There's something else a capsule is used for. It's going to stick to the host tissues. Or it could stick to environmental surfaces. So a capsule has 2 purposes: stick to host tissues and help avoid phagocytosis. What's the endospore for?
Dormant stage.
Yeah, it's a dormant stage. Helps the bacteria get through harsh conditions. The endospore will persist in harsh conditions. When conditions are better, it turns back into bacteria. Good. Alrighty. Okay, you know that one. You know what a basic stain is. It's positive charge, so it sticks to the cells because they have a natural negative charge. It's attracted directly to the cells. And you know what an acidic stain is. It has a negative charge, so it's repelled by the natural negative charge of cells. Okay, when we looked at Klebsiella pneumoniae, what special structure were we looking for? Let pneumoniae be your clue. Klebsiella pneumoniae, what special structure were we looking for? How does Klebsiella cause pneumonia? It sticks in your lungs. It has capsule, right. Okay, we looked at Bacillus subtilis, or we could have looked at Bacillus megaterium, or we could have looked at Bacillus cereus, or we could have looked at Bacillus anthracis. What special structure were we looking for? What are the two endospore forming genera? Bacillus and Clostridium. So if we're looking at Bacillus, what are we looking for? Endospores, right. Clostridium is the other endospore forming genus. Okie dokie. If we're looking at Spirillum volutans, what structure are we looking for?
Flagella.
Flagella, good. If we're looking for acid-fast bacilli, what kind of bacteria are we looking for? Mycobacterium, tuberculosis, or leprae. Or nocardia would be an option. Okie dokie. Okay. When we did our acid-fast stain, if the cells came out green, what does that mean? It's acid-fast negative, or I call it non-acid-fast. Well, what does that mean?
That they are not acid fast.
Right, they're not acid-fast. They do not have the wax in their walls. What's the name of the wax? Mycolic acid. So if it's non-acid-fast or acid-fast negative, it does not have the waxes in the wall. So, what was the example of our non-acid-fast bacteria on our AFP slide? E. coli, good. What color is it going to come out if it does have the waxes?
Red.
Reddish, it's a pink. It's technically pink. On your test, I'll use the word pink, but some books will say red. Okie dokie. When you do a Gram stain, we just did this in lecture today. What if the bacteria come out pale red?
Then it's gram positive.
No?
Gram negative.
Yes. And what does that mean if it's Gram negative?
That it has
Endotoxins?
yes.
And a thin wall. Now, Gram-positive bacteria can actually have exotoxins and endotoxins. Gram-negative bacteria only have endotoxins. I know it's a little weird and confusing, but just keep in mind, Gram-negative, that's endotoxins. Gram-positive, they have exotoxins, but they can also have endotoxins. Okay, let's see. So, what if in a Gram stain it comes out purple?
Gram positive.
Gram positive. Good. Thick wall. Okay. What do you know about E. coli?
Green.
It's Division 1, so it has a thin wall, right? It's enterobacteria, comes from your digestive tract. That's where it gets the name coli. It's also non-acid fast, so it's going to come out green if you do an acid fast stain. And what color is it going to come out if you do a gram stain? Red. And what's the shape of the cells? Rod shape, tiny bacilli, yep. And if you see it on a Petri plate, what color is it going to be?
Like yellow.
Whitish. It's going to be white on the Petri plate. Not white, opaque, bright white. It's going to be like a creamy, translucent white. Okay. What if I asked you, if I did a gram stain of Staphylococcus or Streptococcus or Micrococcus or Enterococcus, what color is it going to come out?
Red. No, purple.
Purple. Why?
Gram
They're
positive.
gram positive, right? Gram positive cocci. Cocci or gram positive, they're going to come out purple. Good. How about our two endospore forming bacteria, Bacillus and Clostridium, and Lactobacillus? They're thick-walled, larger bacilli. They're also gram positive. How about your little rods like Escherichia coli and Enterobacter and Shigella and Salmonella and bacteria in the family Enterobacteriaceae? They're Division 1 gram-negative thin wall. They're tiny bacilli. I'll get you some props in a minute. Okay, don't forget your shapes and arrangements of bacteria. Streptococcus, Staphylococcus, Diplococcus, Diplobacillus, Streptobacillus. There's single bacilli, there's cocci, there's spirillums and spirochetes, right? Okay. Okay. Don't forget the cellular morphology and the colonial morphology. Right? I told you to look them up. Need to know the cellular morphology. That's the shape and arrangements of the cells for Staphylococcus epidermidis, Micrococcus luteus, Bacillus subtilis, and Escherichia coli, and Serratia marcescens, because those are the 5 we've looked at in lab so far. A lot of times you'll get a question about what's the cellular morphology, what's the colonial morphology. The answer is usually easier than you think. You're thinking, oh crap, I didn't memorize the cellular and colonial morphology of all 5 of those bacteria. I'm never going to remember. The answer is in the name. If it's coccus, it's round-shaped. If it's Staphylococcus, they're round-shaped in clusters, right? If it's luteus, it's yellow. Remember, Serratia was supposed to be red pigmented? So on the Petri plate, it would have red colonies. Yours just started to produce red. It wasn't completely red. So a lot of times, the answer is already there. It's not too hard to figure out. Remember, Serratia are the red bacteria. Micrococcus luteus are yellow. Staphylococcus aureus. Aureus means gold. And you saw some of them on your Petri plate. Okay, did that one. Well, I think I have the same question in there twice. With different options for the answer. Huh. Okay. Remember, we talked about fermentation. And when Dr. Pasteur was looking at wine, if the grape juice turned to wine, that was alcohol fermentation. If the wine turned to vinegar, that was acetic acid fermentation. In gangrene, the gas gangrene gives off butyric acid. That's butyric acid fermentation. When we looked at yogurt, the milk, lactose, turns to yogurt, which is tangy.